Welcome to Farmscape Online. I'm Bruce Cochrane. My guest is Dr. Vahab Farzan, a research scientist and adjunct professor in the Department of Population Medicine and the Department of Pathobiology at the Ontario Veterinary College with the University of Guelph. As part of research being conducted by scientists with the University of Guelph and the University of Manitoba, with funding from Swine Innovation Pork under the Swine Cluster 4 research program, are conducting a control study of post-weaning diarrhea on Canadian swine farms. First of all, what inspired this effort and what are the objectives of the work?
This is a project that addresses a very significant problem on Canadian swine farms. And this is basically in a period when pigs, they are very susceptible to different diseases because they just separated from a very, very good environment that they had for 3 weeks with sows, and now they moved together to a new condition mixed with other pigs. So they got stressed because of changing the diet from liquid to the solid diet, and so the whole body is under pressure for some time during the post-weaning period, and that's when bacteria like E. coli, enterotoxigenic E. coli, viruses, and other bugs, they can dominate and make the pig sick. Also, it can cause by other factors like nutrition. This project in particular is focusing on the post-weaning diarrhea, which is associated with enterotoxigenic E. coli. So the goal is to investigate the prevalence and frequency of the disease on swine farms across Canada from west to the east to discover what does exist on swine farms, because there are different types of E. coli. So number one, we would like to know what types of E. coli they cause the disease on swine farms in Canada. Number two, we would like to understand the frequency or prevalence of the susceptibility, the genetic susceptibility to the disease in Canadian pigs. The third goal of this study to look at the gut microbiome, because based on the science, there is a very strong relationship between the gut microbiome and the gut health. The post-weaning E. coli diarrhea is a gut health issue. So if a pig has a very healthy bacterial population in the gut, she will be more likely to fight against E. coli infection. Now, the vaccines are commercially available for E. coli post-weaning diarrhea, and it can be used in sows and in piglets. But one major question is the vaccine should have the antigen or should address the E. coli type that already exists on that specific farms. The E. coli type will eventually be used by vaccine, manufacturer, vaccine company. It will be used to, put something in the vaccine that exactly targeting those E. coli type that exist on swine farms. For genetic susceptibility, I think the information we get from this study can be used by swine breeding farms like CCSI and other swine breeding improvement program to breed the pigs that they are genetically resistant to E. coli. All of those information will be communicated with producer, with the swine industry in different venue. Everyone can take a piece of it and then go from there.